so far we've been talking a lot about how genotypes can lead to certain phenotypes, and if you know you got at least one copy of the dominant allele, you're going to express the dominant trait. But what I want to kind of zoom in on now is how exactly it is that certain alleles lead to certain traits, or certain genotypes lead to certain phenotypes. What is it about these different traits that is the result of the genetic information in each of these individuals? What is it about the combination of alleles that actually results in a black dog versus a blonde dog versus a brown dog? How do you think these traits are the result of, say, 2 copies of the dominant allele versus 2 copies of the recessive allele? Let's go ahead and zoom in all the way into the nucleus of the cells of one of these individuals. Gene expression is the process by which genes are converted into a physical product. And it's going to be a 2-step process: transcription and translation, and then folding into a protein. This is going to be the focus of Unit 3, so we won't get into the details just yet of these 2 steps. But basically, gene expression is taking a nucleic acid, DNA, and converting it into a protein. So taking one type of macromolecule and creating another type of macromolecule from it. And so when we talk about having a certain combination of alleles, we're talking about having a certain sequence of A's, C's, T's, and G's that codes for one protein versus another. When we think about us being a diploid individual, remember that we have 2 copies of every chromosome in our cells. And so we have 2 copies of every gene in our cells. And the alleles are different versions of the same gene. So remember, an allele being a different version of the same gene literally just means a slightly different sequence of A's, C's, T's, and G's for that gene. So with our dog hair example, if dog hair was coded for on this chromosome, the same gene exists on both chromosomes. And if this happens to be, let's say this is a homozygous dominant dog, then on this chromosome, there's A's, C's, T's, and G's that code for a black protein, and on this chromosome, there's A's, C's, T's, and G's that code for a black protein, and that ends up resulting in the pigment of the dog being black in color. Or if we bring it back around to PTC, using the same compound here, but let's imagine different people. So these are not Charles and Karen, these are just random people. Let's imagine a dad who is a homozygous dominant carrier for PTC, meaning that dad has one allele that codes for a protein that can taste PTC and another allele that codes for a protein that can taste PTC. Versus mom, who let's say is homozygous recessive, she's got one allele that codes for a protein that cannot taste PTC and another allele that codes for a protein that cannot taste PTC. If that mom and that dad have kids, let's do the cross. If we have a homozygous dominant and a homozygous recessive person being crossed here, we're going to end up with kids who are all what? Yes, heterozygotes. And so if we think about the gene expression in those kids have 2 copies of the gene for the PTC receptor. They have one copy that codes for a protein that can taste PTC, and they have one copy that codes for a protein that cannot taste PTC. So they've got a certain combination of A's, C's, T's, and G's here that create a protein that is this shape, and a slightly different combination, I'll just write these in a different order for example, of A's, C's, T's, and G's here that create a protein that is a slightly different shape. And again, we'll get into the exact mechanism of this process in Unit 3, but for now, just keep in mind that different alleles code for different proteins. Different alleles code for different physical products. And so if we want to kind of summarize this whole family here, you can have a family with mixed genotypes and mixed phenotypes. And if we zoomed in on their DNA, they are either, like dad, making 2 copies of the same protein, or like mom making 2 copies of the same protein, but the recessive version versus the dominant version, or like the kids making 1 copy of the recessive and 1 copy of the dominant. And remember with PTC specifically, this was an incomplete dominant scenario. So dad, when he eats bitter foods, he finds it really awful, super, super bitter, maybe even spits it out. Mom, when she eats bitter food, she does not taste bitter at all. And the kids, when they eat bitter foods, they can taste it, but maybe it's not that horrible. They've got that kind of mixed phenotype between the 2 ends of the spectrum. All right, let's try a different example. Let's try another incomplete dominance example. And let's put this into the realm, instead of just, straight traits, let's put this into the realm of Mendelian disorders. So traits that can be inherited through time from one generation to the next. Perhaps something that you might have heard of before, like achondroplasia. Achondroplasia is not limited just to humans. It's a form of dwarfism that results in a reduced stature phenotype, and we see it in almost all species of mammals and also a lot of species of birds. And in humans specifically, and in the US specifically, about 1 in 40,000 people in the US has achondroplasia. The majority of the population of humans carries 2 copies of the wild type allele. To be wild type just means most common. The most common trait in a population isn't necessarily the dominant trait in the population. And so the majority of people in the US or globally do not have achondroplasia. The majority of people possess 2 copies of an allele that is creating a bone growth receptor gene that is waiting for an environmental cue to turn off. And so the majority of people have this allele that creates this protein sitting in the membranes of our osteoblasts, sitting in the membranes of our cells that are building bone. And they are allowing for bones to grow all through gestation, all during in utero development. They're also allowing our bones to grow all through young adolescence into early adulthood. And then around, somewhere between 18 and 22, they are activated by something external and they turn off. And so, let's say around 20 years old, these turn off in most people. So your bones grow and grow and grow until you're about 20, and then your bones stop growing. You reach your adult height. Some people possess a mutated version of this gene, a different allele, that instead of producing a receptor that is waiting for an external signal, instead produces a receptor that self-activates. Instead produces a receptor that turns itself off from day one. And so from day one, if you have this allele, your bones do not grow. From day one, if you have this allele, your bones, instead of increasing in size, just don't accumulate any mass at all. If we think about somebody with achondroplasia, if we think about somebody who has reduced stature, and if we assume, like I've told you, that achondroplasia is the result of an incomplete dominant inheritance pattern, then I want you to go ahead and think about which of these scenarios is most likely representative of somebody who is achondroplasic, representative of somebody who has the achondroplasia phenotype, the reduced stature phenotype. Remember, all humans are diploid. All humans have 2 copies of every gene, some combination of 2 alleles, and their options are either to have 2 copies of the wild type allele, 2 copies of the mutated allele, or heterozygous 1 copy of each. So keeping in mind that this is a incomplete dominant scenario, and keeping in mind what you know to be true about these 2 proteins, go ahead and predict for me which of these 4 options you think best describes a person with achondroplasia. And I'll pop this question just below this video.